Hi and welcome to this fifth video in the series about genes and genetic testing, and today we're going to be looking at some more exam questions. Okay, so this question here, question 7. Cystic fibrosis is a life-threatening condition that can affect many different parts of the body. It's a recessive genetic trait. Genetic screening can be used to test for the presence of the recessive alleles. Person found to possess recessive alleles is called a carrier. The diagram below illustrates the risk analysis following the screening for recessive alleles of a couple who are planning a pregnancy. Okay, so suggest why cells from the mouth swabs or blood samples are used rather than gametes. And so obviously, the most obvious thing is the fact that to take swabs from the mouth, or to take a blood sample, is reasonably straightforward, whereas to take a sample of eggs or sperm is less straightforward than that. You can also get a large amount of DNA from each sample. So, for instance, taking blood, you can take a reasonably large sample, and the mouse swab, you can get a reasonably large amount of DNA. If you were to take a female's egg, we're possibly only looking at a single egg, and therefore the amount of DNA you get from that is quite small. Gametes only have half the genetic information, and so therefore the body cells, or the mouth cells and the blood cells, have the full complement. The fact that these cells are genetically identical, and therefore the gametes are not, and so they're all different, and so that you can't guarantee that within a gamete that it's going to actually contain, if you're heterozygous for a condition, you can't actually guarantee that the gamete will be or contain the damaged or the recessive allele in this case. Okay, explain why it's necessary to test for several different recessive alleles in screening for cystic fibrosis. And so cystic fibrosis can be caused by a number of different possible mutations, and so therefore you need to test for those, and therefore if you only test for one, you might miss a subsequent one and therefore have a misdiagnosis. Part C. In the risk analysis shown, if neither partner is a carrier, then it's considered that the chance of having a child with a cystic fibrosis is low. Explain why the probability of having the child with cystic fibrosis is low and not zero. Well, first of all, you might have an incorrect diagnosis. You might have a false negative at that point, so you might have an incorrect diagnosis. You've also got some of the ideas about the fact that not all the possible mutations have been isolated or identified that cause cystic fibrosis, and so in a very, very small percentage of cases that can crop up. The idea that the mutation can occur within the gametes, and therefore the idea of a mutation in both sets of gametes, the sperm and the egg, is also relevant here, and the idea that the mutation might actually occur post fertilisation, and so the child, although its parents may not be carriers for the disease, could actually randomly mutate and produce this the condition post-fertilisation. D. In the risk analysis shown, if one of the partners is found to be a carrier, then screening for cystic fibrosis may be offered to other family members. Explain why the screening is offered to other family members. Well, for instance, they want to know whether they're a carrier, and therefore they can make an informed decision about the action they want to take in the future. So whether they want to have children, or whether they want to check that the person they're having children with might also be a carrier. And finally, E. Using your knowledge of monohybrid crosses, calculate the probability of having a child with cystic fibrosis if both partners are found to be carriers. Draw a genetic diagram to help explain how you calculated this probability. So, I would write out, first of all, like you would do with any monohybrid cross, write out the F1 phenotype, and so the two individuals are carriers. Therefore, you'd underneath that put the F1 genotype, and so for a carrier big C, small c, you might use F for this, for instance, but you write down the genotypes, and that 1 there signifies what you get marks for. So writing down the genotypes of the two parents, get your mark. Identifying what's in each gamete, so identifying that in a gamete you only contain one of the alleles, that gains you a second mark. From there, writing out the Punnett square of the different possibilities, that gets you a third mark. From there, you should identify the F2 genotypes, and so we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. From there, you should be mentioning the phenotypes, and so that means 1 completely healthy, 2 carriers, and 1 sufferer. That gains you another mark, and therefore the probabilities involved here: 25% for completely healthy, 50% carriers, and 25% sufferers, and so that 25% gets you the final mark.